Got it. Okay, perfect. Thank you. Okay, so the purpose of this session is really to help. This was put together specifically to guide people who were recently funded and have these new kind of data standard and requirements to use the BRAIN initiative supported archives. And so this is to help you as a neurophysiologist use DANDY. So which includes conversion to Neurodata Without Borders. So I'm going to go ahead and share my screen. I'll share just this. Okay. So if you've never been to the DANDY archive before, just go to dandyarchive.org, and that'll bring you to this screen. And here you can search all of our available data sets. I mean, we have already 145, we call them DANDY sets, across over 300 users, hundreds of terabytes of data. And if you search, for instance, NWB here, you get all the NWB data sets. And so here's just an example of what the landing page of each data set looks like. We extract some data from the NWB files, and we allow you to have name and description to publish these DANDY sets. So in this first step, I just want to show you basically how you can explore the DANDY archive, and you can put other things like, mouse data. So this is NWB data that has mice. So here's an example of a recent submission from the Allen Institute, and you can see that it does pupil tracking. This is some metadata that we've been able to pull out of the NWB file. So I just want to give you a sense of how to navigate through here. And if you're interested in a particular data set, you can click download, and it will give you the commands to download this data efficiently. This will give you the whole data set. Or if you just want to look at an individual file, you can go into Files, navigate through here, and click Download for an individual file. So that should give you enough to kind of get started in looking through what data sets are already in the DANDY archive. And encourage you, all of these data sets are public. You're free to use them, and we encourage you to use them. We're really trying to promote data reuse. So initially I wanted to show you basically how to do that. But the real purpose of this is to show you how to create an NWB file and upload it to the DANDY archive. So to do that, I'm actually going to use a Jupyter hub that is associated with the DANDY archive, so you can follow along too. So if you want to use these resources and follow along yourself, first you'll need to sign in. So you're going to need to go to dandyarchive.org and sign in and use your GitHub login to do so. And once you do that, you'll be able to go to hub.dandyarchive.org. I've already signed in, but when you sign in for the first time, you'll get an opportunity to choose what kind of server you want to launch. You can just launch the normal one. And then we're going to be using a notebook that's in DANDY Notebooks, DANDY User Guide Part 1. So if any of you are interested in following along, go ahead and launch up that notebook, and you can do this too. We're going to do all of the NWB creation and DANDY upload within the Jupyter hub. You don't have to do it within Jupyter hub. You can do it from anywhere. You can do it from your own computer. But I'm going to use the Jupyter hub just because it's an environment that everything is already installed and anyone can use it. So feel free to, I know I went through that very fast. If you have any questions about how to get started with this, feel free to pop them in the chat, and one of us can help you get sorted out. Actually, there are not that many of us, so feel free to interrupt.
Sorry, can I interrupt? I didn't quite follow that. I'm logged in with the GitHub login, but then I was sort of doing that and didn't quite follow how you got in.
Hub.dandyarchive.org.
Yeah. Oh, hub. Okay.
Yeah.
Okay, cool. I'm catching up. Sorry, I wanted me to sign in.
Yeah, no problem. No problem. Okay, anyone else trying to follow along? You're welcome to just watch, and this is being recorded, so you can look at the recording once it's up.
Okay.
Okay, but I very much appreciate, Anne, that you're following along, so please stop me if you need any more help. Okay, so I'm gonna go forward with this this Dandy user training part 1. So first of all, I just want to explain what Neurodata Without Borders, NWB, is. It's essentially a brain-supported format for exchange of neurophysiology data. So the idea is that you can save all of your data and metadata associated with neurophysiology experiments with sufficient metadata that somebody else could reanalyze this data. So this includes, of course, the neural activity itself, but it also includes information about the data acquisition, the experiment design, information about the subject's behavior. And we have an extension library, which I won't go into now, but that allows you to extend the standard to handle just about any data type that could be associated with neurophysiology. And this is specifically designed for different types of neurophysiology data. So extracellular and intracellular electrophysiology, optical physiology, and behavior. So basically, Neurodata Without Borders tells you a specific organization and structure to put all of this data and associated metadata so that other people can understand it. We can be sure that it has all of the necessary metadata, and we can write software around this organization so that we can exchange tools as well. You can go to nwb.org to learn more about NWB. A lot of resources here. Okay, so the first thing I want to do is just show you how to create an NWB file. So unfortunately, it's hard for me to give you a demonstration that would work for everyone because we have people who use Python versus MATLAB. We have people who are doing extraphysiology versus optical physiology, different types of behavior. There are a lot of different types of data that are supported in NWB, and we just don't have time to go into every single one of them. But what I'm going to do is show you a really basic example of the kind of workflow that goes into creating an NWB file. So for this, I'm specifically going to look at a very simple. I'm going to generate some example data for extracellular electrophysiology experiment. And in doing so, I'm going to create all of the necessary metadata that are required in order to store this electrical series or voltage series of data. And I'm also going to store subject-specific information, which is required by Dandy. Okay, so this is using Python, but just be aware that we have a MATLAB library that works just as well. I'm just showing this example in Python because I wanted to use the Jupyter notebook, and the PyNWB is supported in Jupyter. So, all right, so first the imports, some kind of generic imports, and then some PyNWB specific ones. I'm going to put all this in a function called create NWB file. I'm going to use that later. You don't have to do this yourself. You can just put it not in a function if you want. So first I'm going to create the NWB file object. So basically every session of data is in a single NWB file. It sometimes can be hard to determine what a session is, but for most groups, it's pretty straightforward. If you have questions about that, we're happy to discuss that. Oh, I got a question. Oh, that's Anne responding. Okay. All right. So here's some kind of session level metadata, session description, identifier, session ID, lab and institution data, things like that on the NWB file level. And then the first object we're going to add to this NWB file is a subject object. And here I'm going to put the species and the age of the subject and the subject ID. Subject ID is required, species and age are recommended. So here I'm going to create that object and then set it as an attribute of the NWB file.
And can we do other identifiers here also, other than just like species and age, or are we able to add custom ones?
Yes. So there are other fields that I'm not putting in here. There's like genotype and strain and like a generic description. So actually, if you want to look at what those arguments are, you could go to pynwb.readthedocs.io, and we can go down to API documentation. And okay, subject. So here are the arguments you have available: age, description, genotype, sex, species, subject ID, weight, and date of birth. But there are other attributes that you might want to add to subject, and for that you need to build an extension. Or you could try to like kind of smush them into description if they're not really very easily standardized.
Yeah, I'm thinking for myself and basically anyone else who's doing invertebrate neuroscience, like life stage is also very important, right? Because you could have people working on larvae or working on different instars of larvae and pupae, and you know, they're all the same animal, but it's very different, right? It'd be important to include that.
I was just yesterday in a meeting with someone working with C. elegans, and he was saying that age isn't necessarily a very informative metric for them because they age at different speeds depending on what temperature they're in. So actually, life stage is much more meaningful than like the number of days they were alive. So I think, yeah, the closest thing we have right now would be age, and you can represent that in anywhere from seconds to minutes to years, depending on what is appropriate. But I recognize that life stage might not be well reflected by age in some cases. So that's a good point. I think we can look into maybe adding that as an optional field here. We're learning how to expand this to invertebrates. We started with the standard mice and rats, and we're slowly working our way out to encompass more. So appreciate that. Really appreciate that as a suggestion. Thanks, Dominic.
Thanks.
Hey, Ben. When you say build an extension, is it something you'll go over, or there's like a tutorial for it, or is it simple?
There is a tutorial for it. There is a tutorial. I would say it's how simple it is depends on what kind of extension you want to build. There are pretty sophisticated ones. But if you wanted to do something like add a single additional metric to subject, I don't think that would be too bad. So the tutorial we have for extensions right now is right here, extending NWB. This walks you through all the steps of extending NWB. Basically, you're going to use the schema language that we use to define the core specification for NWB, and we're going to define new neurodata types. I can put that in the chat window here. Make sure you copy and paste that into somewhere else, because when this meeting ends, the chat will be destroyed. I'm sure you all know this by now, but sometimes it still gets me. Yeah, I wasn't planning on going over extensions because I really wanted to make sure that you guys knew how to actually upload data to Dandy and interface between NWB and Dandy. But happy to schedule another talk at another point to go over extensions. Or point you to some video resources. I think we might have some for that. Okay, so this is where we start going into actual electrophysiology. So the first thing we're going to do is create a device. And then we're going to create an electrode group. Electrode groups are basically clusters of electrodes within a device. Your device might just have one cluster, so you would just create one electrode group. And here it's a tetrode, so I'm creating 4 electrodes to put in the electrodes table. Now I'm putting all of these at the same position in the brain, which obviously they wouldn't be, but this is just some example data. And then once we create these electrodes, we're going to create an electrode table region. So this is all kind of preliminary metadata work that we need to create an electrical series, which is actually going to contain our voltage data over time. But our electrical series, we need to say which electrodes from our electrodes table are being recorded from. So that's what this electrode table region does. It basically indexes into this table. This creates a 4-row table. And we're just saying, okay, we're going to give you the voltage data from electrodes 0 and 2 from that table. And we could have chosen all of the electrodes, and actually in the majority of cases, your electrode table region will just point to all of the rows of the electrodes table. And it's 0 index, so this is the first and third electrode. Okay, so I'm going to say, okay, this electrical series has a sampling rate, and I'm going to generate some random data. And some timestamps to be associated with that data, and then create an electrical series object, which contains that data and also contains this electrode table region, which indexes into the electrodes table, tells us which electrodes we're recording from. And then I'm going to add that as acquisition to the NWB file. Now, this is just a few of the data types that we support in NWB. And I realized I went through that pretty fast. But what I really want to do is point you to these tutorials, because we have text and video based tutorials now for the different types of imaging modalities, as well as the different the PyNWB and MatNWB APIs, depending on what you prefer. Both PyNWB and MatNWB are able to create valid NWB files, and you're able to to write in one and read in the other. So it's really not a problem whichever one you want to use. It's just a matter of what's more convenient for you. So we have these videos posted on YouTube. So I encourage you to look through the one that best fits your use case and your preferred programming language. It's going to go over the main data types that are relevant for you. It's going to kind of look like this, but it's going to give you a lot more detail into how all of these data types interact with each other and the different options you have, and kind of more into the design of NWB and how you can leverage it to express your data. Okay, so now I want to show you, once you have an NWB file, once you've gone through this and kind of copy and pasted it and substituted in your actual data, how you would interact with the Dandy archive. So, before I go into that, I will say, if you want any help with this, I recognize there's a lot of information here. We have a help desk, and you can post here. This is the Dandy help desk. There's an NWB help desk, which looks very similar. You can post on either, really. And we like to field questions here, because then it's kind of like Stack Overflow. It's searchable by Google. It's publicly accessible. Other people can benefit from the question and answer. So we have people who are experts in the archive and people who are experts in NWB and the whole process monitoring this site. And so we'd be happy to answer any questions you have. Okay, so now I'm gonna generate some data here. So this is the function that I just created up here, create NWB file. So I'm gonna run that to create the function, and then I'm gonna run this cell to actually create that NWB file. So if I refresh this, yep, I have a new data, and I have ECEPhysExample.nwb. So this cell, this NWB HDF5 IO, takes the file path where you want to write the data, and then writes that NWB file object that we created to that location. Okay, now I want to show you how to upload data to Dandy, but there's one more kind of complication here, which is there's a staging Dandy archive. So the thing is, every time you create a Dandy set, you create this permanent ID, and we basically don't want, in a tutorial where people are kind of playing around with this, we don't want them to create dozens of IDs that don't actually lead to real Dandy sets. So what we did was created this staging Dandy archive, which is identical to the normal production Dandy archive, but you're free to just kind of publish junk data sets there and play around with the tools. So you can see this looks exactly like the normal archive, but we're actually at GUI-staging.dandyarchive.org. So here you'll need to sign in, and then I'll walk you through. I'm going to walk you through now how to actually create a Dandy set and upload this NWB file to Dandy. So once you sign in, you can say new Dandy set, my tutorial Dandy set, create Dandy set. Okay. Register. Okay, so now we have a Dandy set. We have a place where we can deposit data. And you can see here, there are 0 files in here. It automatically made me a contributor, but you can change that later if you want. You can add more contributors later. And And yeah, I'll show you how to add data to it. Oh, another thing we're going to need is our API key. So the way to get that is you click on your little icon here, and then you press this copy API key to clipboard. Okay, and now I'm going to launch a terminal so that I can actually interact with the Dandy archive. Okay, so first thing I need to do is set my Dandy API key. So I'll do that in terminal. So let's set Dandy API key and get the API key from here, paste it. Okay, and yours is going to look different, but it's going to look something like this. And then you can run echo $Dandy API key to test that that this has been assigned as an environmental variable in your session. Okay, the next thing we're going to need is the URL of the Dandy set landing page that we just created. So I'm going to copy this and Dandy download that URL. Oh, I should say, since I'm doing this on Dandy Hub, the Dandy library is already installed, but if you're doing it on your own computer, you would first do pip install Dandy. That'll take care of that. Okay, so Dandy download that Dandy set. All right, which will create this folder here. And inside of it is your dandyset.yaml, which gives you already metadata about the Dandy set, which there isn't a whole lot yet, but it does have, you know, your example citation. It's got the title and description that you've already given to it, and some contact information. Okay, so now let me just move this over here so we can see both. All right, so let's CD into that folder that we just created. Okay, so now what we want to do is organize the data that we just produced. So let me show you. So we have our data in here in this data folder, and we want to organize that in our new Dandy set folder. So Dandy has a particular naming scheme that it uses for each file that's kind of inspired by bids. I guess it's bids-like. So what it does, when you run Dandy organize, it will go into each NWB file, and it will parse out the necessary metadata that it needs to rename these files according to the Dandy rules. So so I'm going to Dandy organize data fdry. So that just kind of parses through all of it without actually moving anything, and then do it without fdry to actually organize the data. All right, so now if we go into here, we have this new subject 001, and then our Dandy set is within here. So this, it actually doesn't create copies of the file, it actually creates soft links. So it creates a new organization without copying the data itself. But this is the organization that's going to be uploaded to Dandy. And then now that we've confirmed that we have something in here, we can run Dandy upload. And then we have -i Dandy staging, which we use just because we're operating with the staging archive, not the standard archive, not the production server. So we do that. All right, so it has now uploaded the data, and we can go to our landing page and refresh it. And there we go. We have data in here. It's automatically pulled out several variables. It can tell that we're using NWB. We have one subject. We're doing electrophysiology on a mouse. All of this is pulled out from the metadata in the NWB file. We can also look at the individual files and navigate and download or delete or look at the metadata associated with each of these files. All right, so if you wanted to add more data to an existing Dandy set, that's actually really easy to do. Here, I'll show you, I'll create a second NWB file where I'm just going to change the subject ID. And the electrophysiology data is randomly generated, so that's also going to change. So here I'll show you, if you go to data, we have a second NWB file here. So I'm going to run organize again, pointing to that data file, a data folder. Okay, and then after we run organize, now we have two subjects here. And you can run dandy upload again. And so that's going to intelligently look at what files are exactly the same locally and on the server, and it's going to skip them. So you can see it skips this file because it's already there. And this file is new, so it uploads it. And if I were to change this, it would see that it's changed and it would upload the new version of the file. And so now I can go back to the My Dandy Set landing page. And okay, so we've increased the size of this file. We have two files in here. And if you go to the Explorer, you can see that this new NWB file has been added. Now, the one thing that we are missing to actually publish this, it's in draft mode right now. If we want to publish this, then we need to go into metadata. And here you can add all sorts of great metadata, like the contributors. So that's basically authors of the Dandy set, funding, resources. You can link to ontological terms associated with certain anatomical regions, which makes this far more searchable. But the one thing that's required in order to publish that we don't have yet is a license, which tells people how it's allowed to be used. I would probably recommend for most people CC BY, which means you can use it, but if you use it, you have to say that you used it. You have to acknowledge that this is that you used this particular Dandy set. And you can check out creativecommons.org to learn more about what these licenses mean. That's kind of the gist of it. Okay, so I saved that. And now if I go back to the home screen and I refresh it. All right, so it actually takes some time to process this. I'm going to come back later, but this will be publishable once Dandy recognizes that that all of the metadata is taken care of. Okay, so that's basically how to create an NWB file and how to upload it to the Dandy archive. I just wanted to show you some other tools that are available on the Dandy Hub. So you can use a tool that we've been working on called NWB widgets to render an interactive visualization of the NWB file. For this particular one, it's not that exciting because it's two channels of randomly generated data. But you can also do this on files that have already been published on the Dandy archive. So this is a segment of code that where I'm directing to specific NWB files, in particular Dandy sets of real Dandy sets that have already been published. And so basically here, we're using the Dandy API to identify where the S3 location of that NWB file is. And we're passing that into PyNWB and into NWB widgets in order to produce an interactive visualization of that published NWB file. The cool thing about this is since we're pointing it to an S3 location, we're not actually downloading the entire file locally. We're just kind of streaming data as needed as it's being visualized. So we can go into acquisition and you can see a number of different time series data that are being recorded here. This is taking a little while. Okay. So this is pulling the data directly from the NWB file. Here we have a trials table, which tells you the start and stop time of each trial, whether it was an error, whether they ran right or left. So this is kind of a figure 8 maze. If we go into the behavioral data, you can actually see the figure 8 of the maze. So let's go into trace view. Okay, let's look at 50 seconds of data. All right, so you can see that's one run through the maze. And if we made it 100 seconds, okay, here's a full left-right run through a theta-shaped maze. And we also have neural activity. You can look at the units per time. So this is how to explore NWB files in a code-free way using NWB widgets. But you can also just use PyNWB. We've created the NWB object here. We pass that into NWB widgets, but you can access that NWB widget. You can access that NWB object directly and pull out specific data and do your processing here right in the notebook. So this is an example of pulling out the first 1,000 time points of position sensor 1. So this NWB widgets, this whole system works for all sorts of different types of data. This is just one example from the Bizaki lab, but you can run it on data from the Allen Institute, from the Giacomo lab. Very large data sets will work because we're only pulling the data that we need to actually run these visualizations. We're not downloading all of the data before we visualize it. So you can visualize data from a 46 gigabyte NeuroPixel file without a problem. So that's kind of the basics of what I wanted to go over. I kind of wanted to transition into more of a Q&A section. But I also wanted to point you to how to schedule a consultation with us if you want more one-on-one help. I'll put this link in the chat. So this is a Google form where we just ask you a few questions and we ask you to pick a time slot. This is set up so that when someone picks a time slot, it is removed from the options. So all of those time slots, you won't intersect with anyone when you pick it. And there we can have like specific conversations about, ultrasound or ant specific data and maybe creating extensions that would work for different specific or wide field specific types of data. But yeah, I mean, I think it would be useful, at least at this stage, if we could have more of a group Q&A, just so we all kind of benefit from each other's questions and answers. So yeah, at this point, I'd like to open the floor and see if anyone has any questions I can help with.
I'll go. So we're using optical physiology, and if I remember correctly, how it's set up is really one data set, one optical image data set per subject. We're going to have more than one. And I guess how would we, even though it's a single session, it's going to have several different time chunks along the way. So for example, we image for 30 minutes, and let's say that we go through the brain sequentially. Each plane that we do takes about 5 minutes, let's say, and then there's a time gap between them, and then another one, and then a time gap between them, another one.
Yeah, that's perfectly fine. You can create multiple time series objects within a session. That's perfectly fine. We don't, I mean, I'm just showing you here in my example, I have one electrical series, but you can just have more, you can have more than one. The one thing is, if you want to call them all a single session, we would need to know the start time of each of those blocks of data with respect to the session start. So basically you need to, keep track of when you started each of those segments of data.
Okay.
But I can go to this table really quick. Let me share my screen again. And see, where's the table here? So write optical physiology data. So we can go to the notebook here. Oh, this is a little bit outdated. Actually, a better tutorial is on the NWB API docs, and I can put a link here. So we've got tutorials and domain specific CAPTCHA imaging. I'll put this here in the chat. Okay, so you create a single imaging. Well, actually, you could create multiple imaging planes if you're progressing through the brain, and then you would create 2 photon series objects, which would contain the actual data within. And these 2 photon series objects, they extend time series and add specific information about that you need for optical imaging, particular linking to an imaging plane, which holds metadata about the excitation, emission lambda, and stuff like that. And then once you have that, you can create a plane segmentation object, which is where basically you would describe parcellation of that image into individual ROIs or neurons. And then you can also add metadata associated with each of those neurons. So if you want to label them as putative cell types or excitatory or inhibitory or something like that, we're pretty flexible in the type of metadata you can add to those ROIs.
Okay, so it sounds like I would have a collection of 2 photon series objects with related plane segmentation objects for each one.
That's exactly right. Yeah.
Okay.
You'd probably want to create for each individual 5-minute segment, you'd probably want to create a separate imaging plane and 2 photon series and plane segmentation table.
Sorry, repeat that.
So for each of your 5-minute segments, you probably want to create a different imaging plane because it's a different place that you're recording from. Sounds like you're recording from a different depth. And then you would want a separate 2 photon series that links to that imaging plane.
Okay.
And then you would also want a separate plane segmentation is like a table where we describe the ROIs. So that's kind of like downstream processing of this. You don't have to do this. You could just share the raw data, but usually people like to describe their own parcellation of that image into putative neurons and then provide the activity traces for those neurons. So you would use plane segmentation to describe the parcellation of the image, and then you would use an ROI response series to store like the DF over F traces for each of those putative neurons.
Okay. Separate question. Is there any, I think I know the answer to this, but is there any C# implementation of NWB?
So here's the thing. NWB is basically like a specific form of HDF5. Now we have, we support other backends, but the primary backend is HDF5. And so we only provide NWB specific APIs in Python and MATLAB. Basically, that makes it convenient to read, and it helps you create properly formatted HDF5 files that follow all the rules of NWB. If you wanted to do it in another library, HDF5 has APIs in all sorts of different programming languages, and presumably they have something in C#. It would be pretty straightforward to read NWB files using any HDF5 library, because after all, they're just HDF5 files with specific structures. Writing would be tricky because there are a lot of rules that you have to follow. And the APIs help you do that and make it much easier to do that. But I think it'd be really difficult to use just a generic HDF5 API and create a properly formatted NWB file. One workaround that might work for you is if you create a template file using MATLAB or Python, and then you could have kind of minimal code to add a specific data set or something like that to that file using whatever HDF5 API you want to use.
Okay.
And we can, you know, schedule a meeting to talk about exactly like how you would generate that file and what, you know, how you would do that. Happy to help with that.
I would schedule something like that in the with the forms that you showed at the bottom.
Yeah, if you could use the forms, I think that's the easiest way to do it. I guess I organized this thinking that more people would show up. So I suppose, you know, we could, since there aren't that many, we could probably just figure out a time. But the forms are probably easiest.
Yeah, I'll go. I'll do that.
Ben, we're planning to do some multi-plane imaging also, just like switching between like two planes interleaved between each other, right? Like during one trial. This isn't something that we're doing yet, but it's definitely in the works. So can you just talk a little bit about how that should be handled?
Yeah, I mean, I guess it depends. You know, it's pretty common for people to have like multi-plane imaging where they're switching so fast between the planes that they're basically doing volumetric imaging. Is it that kind of thing, or is it like 5 seconds of this layer and 5 seconds of this layer kind of thing?
No, it's very fast. Yeah, but okay. I mean, it's different planes for sure. But
so it's like volumetric, essentially, like the beginnings of volumetric imaging, where it's like you have a z dimension, but it's just 2 layers. Is that right?
Correct.
Yeah. So so I guess I should say the names of these objects were determined before multi-plane imaging was a thing, but you can use them for multi-plane images, no problem. You can create an imaging plane. I mean, it's a bit of a misnomer, but you can describe it in 3 dimensions instead of 2. It's really an imaging volume, I suppose. And then you can have, you know, GCamp or whatever excitation in 3 dimensions over time. That's perfectly fine. You can describe your ROIs across a 3rd dimension as well. I guess one thing that I would distinguish between is whether you're thinking about this in terms of like an ROI can exist across 2 planes or not. So sometimes the planes are so close together that you're really doing volumetric imaging and you're identifying ROIs in a volume. And sometimes the planes are further apart, and you know you're really doing like planar segmentation on one plane and then planar segmentation on another. And in that case, it might make more sense for you to have just 2 plane segmentation tables, one for each, you know, one for each plane of the image. Kind of depends on how you're thinking about it.
Yeah, I think for us, it would most likely be completely different ROIs in different planes. But this is still down the road a little bit. So it's maybe once we get started, we can touch base again and see what's the best way.
Yeah, absolutely. I mean, it's very doable. I mean, I would recommend that you work through this OPHIS tutorial I'm pointing to, and I mean, you'll see the way that we associate data with metadata is pretty modular. So you'll kind of get a sense for where you can make duplicates of objects and store an additional plane.
Thanks.
No problem. I mean, I guess we're coming up on the hour, so I'm happy to answer more questions, but I'm also happy to kind of do things more individually.
I guess the other remaining question for me is, I was talking about those imaging planes and then associating behavioral information with it that is We have, I guess it's, well, I don't want to say three-dimensional, but it has angles and positions of different, what's a good way to put it? Oh, I guess just generally to say how to attach behavioral data to the imaging data.
Yeah. Yeah. So let me go to, all right, so this is imaging specific, but let me just go to tutorials. And NWB basics. Let's see here. Where's the spatial series? Where do we move that to? Ryan, do you remember?
I saw it at the bottom of the calcium imaging tutorial.
Oh, was it? Let me see.
So yeah, it was part of our hackathon tutorials. I don't know if it made it in here.
I see. Okay. So we can just.
Oh, sorry, I was in OFIS, not calcium.
Oh yeah, no, it's fine. I think we're working on adding this tutorial to the PyNW website, but right now you can find it here.
Yeah, you can change hack 8 to hack 10. That's probably, or 11. Try 11.
Hey, okay. Yeah. So
That's
this is
the
a
latest
little more
one.
comprehensive. So it has spatial series and position here. So it's basically like additional objects that are part of, attached to the NWB file like completely separately from the imaging objects. So you create a spatial series object, and this is meant to store like the position of the animal in space. I recognize you might have like more complex, we'll have to talk if you want to do something more complex than like position and orientation, but that's basically the default data types that we have available for behavior. And yeah, you add this spatial series, you create timestamps for it. So the way that it's kind of cross-linked with the imaging is really simply by the fact that you give them timestamps that are in reference to the same session start time. So basically, because of that, then someone can go in and you do their alignment, take like a behavioral event and pull OFIS that are associated with that behavioral event, because they all have timestamps that are they don't have to have the same sampling frequency. They just have to have the same starting point.
Okay. Yeah, I guess we do have a little bit more complex data in that we do zebrafish larvae and then we're tracking tail movement. So we have, let's say 20 tail positions. Each one has an X and a Y and then an angle associated with it. So there's, I guess that would be 60 data points per image that we have, and then the time series that expands from there.
Yeah. So we've been working with Deep Lab Cut to create an extension that works for their kind of complex behavior output. But that still has XY positions and not angles. So this allows us to have multiple joints in most cases that are connected somehow, and we can track the motion of each joint. I think you'd probably want something similar to this, but honestly, I think it might warrant its own extension similar to this, but something that could also include angle.
Okay, yeah, it's possible to recalculate it. It just seems like that's part of the processed data.
Oh, I see. So you can calculate the angles by knowing what the other joint positions are?
Yeah, it's like sequential angles between the positions. So
I see.
you have a straight tail, they're all along the same.
Then I think I think it might make sense to use this. I guess it would just kind of require a little helper function where that can easily compute the angles of everything.
Yeah. All right.
Yeah. Yeah, so this might work for you. I mean, you might want to give it a try. Essentially, this allows you to kind of group spatial series together into a single like pose estimation object, which allows you to track multiple points of an animal. You name the point, and then you give the data for the point. And so you can have multiple different joints here. So this is for like left paw, this is for right paw, for instance.
Going back to the spatial series, it says that data is n time by n feature. That seems like it would be suitable, meaning like we have each time point along the.
Yeah, but it's like time by x, y, z. So it's either time by 2 or time by 3.
Okay.
And when you're doing x, y, or x, y, z. It's not generally used to track multiple different joints simultaneously. That's like you would use multiple spatial series objects for that. And pose estimation series is just an extension of spatial series. It has a little bit of extra information about like how confident we are in the estimation of the position of this joint at a certain time.
And this could be, I guess this could also be an area where an extension is written for us, or that we write an extension.
Oh yeah, you could. It's I just, we're always trying to find opportunities to find synergies where extensions that are written for one group work for another. And it sounds like that might work in your case. So I would want to try this first, and then if there are certain limitations,
sure,
we can figure out what changes we need to make.
All right, that sounds good. Yeah. Thank you for the presentation too.
No problem.
Ben, so I have a question.
Great.
So I take a look at the training video, and it seems that the spiking time can be stored in the NWB format. But how about the spike waveform? If we would like to keep archiving those snippets, and do you think that'd be doable?
Yes, that is doable. So we would recommend that you just put that in the units table. So if we go to the go to the API documentation. This is in miscellaneous units. All right, units is the units table where you would put where you store your spiking data, and then wait, is waveform not in here yet?
That's weird.
Waveform not in here?
That should be in there. I'll double check.
Well, it's not documented, but it is there. Yeah, so you would put this basically every row of this table is a different unit, but it can contain the rows can themselves be vectors. So spike times is a vector where each row is a vector, and then waveforms similarly you would have like a matrix of that's like number of times by number of time points in a snippet. And I'm happy to work with you to show you how to get those into the table.
One other thing, since we're talking, I started a new Conda environment to try the Dandy CLI.
Great.
And I got an error.
If install Dandy gives you an error?
So it was Dandy download and then the URL, and then it hands me a error type string enum is not an object boolean, and then some more kind of stuff. I guess the
you wanna-
question there is, it isn't so much solving the problem, is this something that I would do from create a pull issue for, or excuse me, create an issue for on
Yeah.
GitHub? Okay.
Yeah, you could create an issue on GitHub. So that would be Dandy CLI.
Okay.
My
Ideally, paste the output as you see
Yes.
what happens, right? With that
the
trace
whole
version
trace
of
back.
it, and
Okay.
the version of the client, especially if you have a little bit dated one and you could afford upgrading it before, just to check that most recent one doesn't resolve it. That would be great.
Do you mean like my Python version, or?
No, just Dandy client. So whatever you run,
Okay.
the version of Dandy client you're using.
Okay.
Yeah, you just do pip install -u Dandy just to make sure you have the most updated version that's released.
Yeah.
But if you just created the content environment, then you probably do have the most updated version. So
Yeah, just to try it out, I just started with a blank content environment, did the pip install u, and then
and you created, oh, okay, so you're trying to download another data set or one that you created?
yeah, going through a tutorial, it said, yeah, download this data set. All right, I'll download this data set.
You created the new Dandy set first and then you downloaded that, or you just picked one at random to download?
No, it was I downloaded somebody else's. It wasn't at random. It was a part of this
Oh yeah, okay.
tutorial.
So did you set your API key first?
No, I didn't set an API key.
For
Yeah.
download, you don't need
Oh, you usually don't need it for download. Yeah. All right. Yeah, we need to do error.
All right, I'll just go into GitHub and create an issue.
That'd be great. Thanks.
Cool.
I have to get going, but thank you very much for the training. I'm sure we'll be in touch later.
Great. Sounds good.
Thank you.
See you, Dominic. All right. Kai, I got a thumbs up from you. Thank you. So Sandhya, how's it going? Do you have any questions?
I think I have a decent understanding. At first, I'm going to be working with pretty straightforward continuous eFIS data, so I think that a lot of that is fairly straightforward to enter in. So I feel pretty confident with that. I'm trying to think if there was anything. I think I just need to go through the MATLAB version and make sure I understand all of that too. But when I looked through it, I felt pretty confident. So this was helpful to see the sort of GitHub-ish side of it, though, and help my understanding of that, because I'm a little inexperienced with GitHub itself.
Yeah, sure. I get that. It's definitely something that I mostly learned after grad school, but I think it's really helpful to get acquainted with it, because it's really a great tool for collaborative development. Yeah. All right. Cool. Thanks for coming.